Hello everyone, welcome to the YouTube channel DocuC, where we explain the more complex topics of ophthalmology in a more simpler manner. So today's topic is embryology. Now let's start with the basics of embryology, the early development. So when a male and female gamete unite, that's called fertilization, and then there's formation of zygote. Once the zygote is formed, it keeps traveling. It's a diploid cell, so it keeps traveling within the oviduct to reach the uterus. So during its journey, it divides and it forms various stages of embryo. So the second one is morula, where there occurs a blastocyst activity. Now, what is blastocyst activity? It is basically formation of trophoblasts, which is the outer layer, trophoblasts, and then the inner layer is embryoblast. That is easy to understand. Embryo usually stays within, so that's why it's the inner cell, inner mass, and this trophoblast is like a protective layer which surrounds the embryoblast. So once that is done, there is a formation of blastocyst. Once this blastocyst activity is done, there's formation of blastocyst. Now blastocyst keeps moving and then it gets implanted within the uterus. So you can imagine the various stages of embryology where these group of cells, they keep traveling and finally enters the uterus and then it gets implanted. Okay, so now what is blastocyst exactly? Now within the embryoblast, like I said, embryoblast is the inner mass. So within the embryoblast, there occurs formation of two cavity. So one is the amniotic cavity, the other is the yolk sac cavity. What divides these cavity into two separate parts is basically this germ cell layer. So this germ cell layer comprises of two layers. One is the epiblast and the second one is the hypoblast. Now what epiblast does is epiblast is future ectoderm and hypoblast is the future endoderm. But we can see there's one layer missing, which is the mesoderm. How's that formed? So now that is formed with the formation of something called a primitive streak or a knot. So in primitive streak and knot formation, what happens is this epiblast, some of it gets detached and then it goes, and so suppose I'm making a schematic diagram of it. These were the two layers, right? We're seeing it in a sagittal form. These were the two layers. Now some of the epiblast detaches and then it goes and settles within there's a potential space between the epiblast and the hypoblast. So then it goes and it settles there. And it settles and it condenses, thus forming something called a trilaminar germ disc. Like how there was a bilaminar, there is a trilaminar germ disc. Now you can see here, this is the next stage where a trilaminar germ disc is formed. Now if you cut this section, this is what you can see. There are three layers, okay, throughout the embryo. Now the next part will be the formation of a neural plate. Okay, so neural plate basically starts at the cranial end, the cranial part. Okay, so once there is a neural plate that is there, the neural plate starts folding on itself to make a neural tube. Now, what is a neural tube? A neural tube is the primitive version of CNS. This is how CNS begins to form, through neural tube. So now this was a basic structure of how the early development happens. Now we'll move on to the ocular development, where our interest lies. Now coming to how the eyeball itself is formed. So like I previously explained, there was a neural tube which was formed. So suppose this is an embryo that we're seeing, like a bird's eye view, and here the primitive streak was formed, and then the mesoderm was formed. After that, it keeps increasing and it goes throughout the embryo. Then, if we see in a coronal section, there is formation of neural tube, which is basically neural ectoderm folding on itself. Okay, so then it starts with one part of the embryo, and then it keeps moving. So then the fold of the neural tube begins at some point in the embryo, and then it extends cranially and caudally. Okay, so it is basically like a hollow pipe which is being formed throughout the embryo. Okay, and ultimately this will close from the above and below. So cranially it will close and caudally it will close. So cranially, when it is being closed, that's when, even before it's closed, there is a hollow diverticulum that is formed. It goes laterally, it grows laterally from the neural tube. So like you can see in this picture, this is basically the diencephalon, which will form the cerebral hemispheres. These you can see, these will form the cerebral hemisphere of the embryo. Here, this area will form the third ventricle. and from its wall is a hollow diverticulum that is being formed, and then it's getting slightly dilated. This itself is known as optic vesicle. Optic vesicle. So what is optic vesicle? It is basically a hollow diverticulum which evaginates from the neural tube, and this occurs even before the neural tube is fused cranially and caudally. So this diverticulum formation begins even before the neural tubes are closed. So, like we saw in the previous picture, there's diencephalon and then there's formation of the optic vesicle. This begins at the gestational age of week 4. Then gradually it keeps extending. This hollow diverticulum, it keeps extending laterally, and then at some point it is covered with, as you can see, this is surface ectoderm. So this was neural ectoderm. This outpouching was formed from the neural ectoderm, and this is the surface ectoderm. Surface ectoderm. So as this outpouching keeps extending and constricting, it comes in close approximation with the surface ectoderm. And when this happens, when there's close approximation with the surface ectoderm, the area lying just in front of the optic vesicle starts thickening. As you can see, it is thickening up. Now at this point, this will be called as lens placode. We will talk more about it. This is known as lens placode. And this particular thing, this is just a clinical nugget that you can remember. The fact that this area is being formed into lens placode is induction. It is an example of induction. Now we will cut this section from here and we'll have a closer look. So this you can see is basically a cut section of this. So now here, this is neural ectoderm, which is basically the outpouching of the diencephalon. This is hollow and it's being connected to the cavity of what will be the third ventricle. Okay, and this, as it keeps, the outpouching keeps growing and extending, there's constriction of this area, and this area is known as optic stalk. This is optic stalk. And now, like I told you here, this is surface ectoderm that comes in close approximation with the neuroectoderm, the optic vesicle, and it thickens up, known as lens placode. Okay. At the same time, there is mesenchyme that is covering the ensheathing the optic vesicle. Now, what is mesenchyme? Mesenchyme is nothing but combination of paraaxial paraaxial mesoderm plus neural crest cells. Neural crest cells, NCC. Okay. So this is what is mesenchyme. Okay. So now the green bit that you can see, so there's mesenchyme that is ensheathing the optic vesicle everywhere except the apex. Why not the apex? Because that's where the surface ectoderm is approximating with the optic vesicle, the apex of the optic vesicle, as it needs to form the lens. So right now at this point there is formation of lens placode. So by week 5 there are 3 major things that happen. So first is there's invagination of the lens placode. So when it's invaginating, the optic vesicle needs to make some space for it to invaginate. So then it starts folding upon itself. So this folded double layered structure is known as optic cup, and this forms the future retina. The outer layer is forms the retinal pigment epithelium, and the inner layer forms the inner layer of the retina, the neurosensory retina. So then this space is made, and then the lens placode has invaginated. So this would form the future lens, the crystalline lens. Second thing to happen is that the mesenchyme that was ensheathing the entire optic vesicle starts to condense. This, you can see, it is more condensed rather than how we saw it was previously spread out. The third thing is there's entry of hyaloid vessels within the choroidal fissure. So now what is choroidal fissure? I'll explain in the future slide. But just remember, there's entry of hyaloid vessels. This is the major blood supply at this point for the eye that has been formed up till now, up till week 5. So you can see there's hyaloid vessels that enter the choroidal fissure and then becomes the major supply of the structure. So these 3 things we need to remember. Besides that, as the lens placode is invaginating, there's formation of lens pit. So basically, if we see lens pit at this point, we know that there's invagination of the lens placode, but it is not completed yet. It is not sealed off by the surface ectoderm. So the presence of lens pit ensures that the invagination has occurred. So by week 7, as we can see, the mesenchyme has more condensed. the choroidal fissure has closed up and the hyaloid artery is regressing. We can also see the formation of the lens. The secondary fibers have appeared at this point, and the surface ectoderm has started taking its shape in terms of lid formation. Okay, so the mesenchyme that is ensheathing forms the sclera. The inner bit, the outer bit is more condensed and compressed. So this is sclera. We know sclera is a tough structure. However, the inner part is more vascular. You can see that it is more vascular and it is looser. This forms the choroid. Another important thing we need to know is that there are three waves of mesenchyme that comes within the surface ectoderm, in between the surface ectoderm and the rest of the structure. So it's important to remember the first wave of mesenchyme that enters is the closest to the lens, and it forms the corneal endothelium. This bit forms the corneal endothelium, and then the rest of it forms the remainder of the cornea. So when the first wave occurs, when the first wave of mesenchyme enters in between the surface ectoderm and the lens, and it lies closest to the lens, this is the corneal endothelium. Okay, and the rest of the two waves, what does it form? It forms the remainder of the cornea, the iris stroma, as well as the iridocorneal angle. So, three waves. The first wave is important to remember. It lies closest to the lens, forms the corneal endothelium, and then the second two waves, the other two waves, forms the remainder of the cornea, the corneal stroma, the iris stroma, and the iridocorneal angle. Okay. The presence of choroidal fissure. What is choroidal fissure? It is basically a deficiency in the formation of the optic cup. Now, when I said that there was invagination of the lens placoid, that is happening somewhere here. So the optic vesicle started folding upon itself to form a double layered structure. But this does not happen equally circumferentially. There is a deficiency in the inferior aspect. Now, why it's present? So that the hyaloid vessels can enter and then supply this structure. Hyaloid vessels being the major supplier of the developing eye, it needs space to enter, therefore, this deficiency remains. However, this also needs to start closing up as the gestation period increases. So you can see this is the coronal section, how the both the layers start fusing upon itself in order to close the choroidal fissure. So again, here you can see the choroidal fissure is closing. These are the vessels. They start fusing in the inferior aspect. Now it begins from the optic cup and it progresses distally and proximally. Proximally being towards the optic stalk and distally being towards the lens. So it needs to start closing up. So this is the significance of choroidal fissure. So now by week 8, we can see a lot of structures have developed. So firstly, we can see that this green part, which was the mesenchyme, has now developed into sclera. And then the inner portion, which was loose and vascular, has formed the choroid. The optic vesicle, which had folded upon itself to form a double layered structure, is now differentiated into retinal layers. Thirdly, the ganglion cell axons, they grow towards the optic stalk. Now you can see these black lines. These are the ganglion cell axons, which are then moving towards the optic stalk. This in future forms the optic nerve. And other than that, you can see the secondary lens sutures are being formed and the secondary vitreous is being formed with the degeneration of the hyaloid artery. So this happens in week 8. We'll come to a few clinical pointers in regards to the development of the eye. Firstly, what are the anomalies that can occur in various points of development? So pigmentation occurs at week 5. So by week 5, there's pigmentation of the iris as well as the retina. If this fails to occur, it is known as albinism. So this becomes a part of a syndrome. As you can see in the eye, there is lack of pigmentation in the iris. Secondly, what can happen is there's failure of closure of the fissure, the choroidal fissure. I had previously explained. So the choroidal fissure starts closing by 3 months and is supposed to completely close by 7 months. Now, due to some reason, if there is failure of the closure of choroidal fissure, something called coloboma is formed. Now, coloboma, as you can see in the picture, this is coloboma that is also including the iris. This is known as a keyhole iris. Keyhole iris. Now, this can either involve the iris, the lens, zonules, and the retina, or it can solely involve just the retina or the optic nerve, depending on what stage of the failure has occurred. So, thirdly, there is something called persistent pupillary membrane. As you can see, within the pupil, there is some sort of membrane that is present. Now, this is due to persistent hyaloid vasculature. Like I had previously explained, hyaloid vessels enter and supply the structures in the eye. Specifically, there is tunica vasculosa right over the lens. Now, if that fails to regress completely, there is persistent membrane like this. These are the 3 pointers that you can remember. These are small clinical nuggets of sorts. So that was the basics of development of eye. I will make more videos and I'll go into more details of how each structure is formed. For now, there are a few mnemonics that I have mentioned in the video, and that will help you to remember what structures from what layer. So coming to surface ectoderm, I would like you to remember the term level. Okay, level. So L for lens, E for eyelid skin specifically, V for vitreous, but only the primary vitreous, E for the epithelium of cornea, L for lacrimal gland. Next is neuroectoderm. Neuroectoderm, we'll remember romes. Romes is R is retina, O is optic nerve, M is the muscles of iris, E is epithelium of iris, S is secondary or tertiary vitreous. And then third, we'll remember mesoderm. So now mesoderm, we'll remember through meso. Okay. So meso is muscles of the motility. That means extraocular muscles. E being endothelium of the blood vessels. S is sclera, only the temporal part. And O is ocular primary vitreous. Now, whatever is left is formed by NCC. So whatever structures are not mentioned above is formed by NCC. So this is a little trick for you to remember what structure is formed by what. That's all for this video. Hopefully this added a little bit to your knowledge and helped you remember ocular embryology a little better. Thank you.